Hello everyone, and welcome to the third pre-panel event of An Evening with Neuroscience 2021, hosted by Gray Matters Journal. My name is Jay,
And I'm Chloe.
and we're your event coordinators for this year.
Gray Matters is an undergraduate neuroscience journal focused on increasing access to neuroscience, regardless of background. An Evening with Neuroscience is our flagship event, featuring interdisciplinary speakers who share their neuroscience knowledge with us in an informal, fun way.
Art imitates life, and so too does neuroscience art allow us to connect to the brains we possess. Today, you'll be hearing from Dr. Greg Dunn, a neuroscience artist whose scintillating, yet scientifically accurate pieces leave audiences in a state of awe around the globe.
We hope you enjoy his presentation, Promising Grounds at the Intersection of Neuroscience and Art.
Hi everybody, my name is Greg Dunn. I am an artist and a neuroscientist, and I'm here to tell you a little bit about my work, about how I got to this spot in my career, which was never the plan, by the way. Things just kind of turned out this way, so it might be a little bit interesting to hear about that. And I wanted to tell you in more detail about one of my projects called Self-Reflected, which I think you will find interesting if you like the combination of art and science, engineering, physics, that sort of thing. Stick around for that. I'm going to start now with talking a little bit about what I think is the true connection between art and science here. And I apologize, by the way, the resolution is low. I know I'm having trouble recording at higher resolution without terrible lag. And anyway, that's where we're at. So I wanted to call your attention first to this picture in the upper left. Alyssa Monk's. This is a photorealistic oil painting, mind-blowingly. And I say this is a science experiment because 100 sufficiently skilled artists would all paint the exact same thing. They're looking at the same data and are trying to replicate it. They're copying something from the external world. This is not a knock on their technique. This is not a judgment call whatsoever. This is simply saying that the point of science is for many different scientists to be able to repeat the same experiment and get the same results. That is also the goal, essentially, of photorealistic painting. Whereas abstract painting, if you tell those same 100 artists, this is a canvas by Gerhard Richter, by the way, who's done both photorealistic and abstract work. You tell the same 100 artists, okay, I want you to paint what it feels like when you feel like you've lost your favorite jacket, or just some random thing, what it feels like when you're angry. All of those paintings are like totally different from one another because they're referencing their internal reality. They don't have anything to fall back on. They're bringing something out of themselves and into reality. I think that defines more the extreme side of art, of art in that direction. And then you have basically everything in the middle. Concept art, as long as we're going to be talking about visual art, is a great marriage of the two. This artist, Craig Mullins, understands light and perspective and composition and all these things that we use to navigate our typical world, but invents a new place that doesn't exist. That's basically what we're doing all the time. We're using our understanding of the external world and our opinions and ideas which come from within us in order to bring something new into our world. And as this relates to the subject of neuroscience art, which is kind of my field, you have seen these images absolutely everywhere, I'm sure. What about these? You've probably seen a few of these too, of neurons. This is what the brain looks like. It's blue. There's little bolts of lightning going through it. And what happens is that artists, illustrators, we're used to seeing these images in our world, and we make some small change upon them. And it's the category isn't changing here. We've seen a bunch of images like this up to this point. I mean, and to be fair to these illustrators, they're trying to get across a very complex idea in a simple way. These are probably some of the other images you may have seen of the brain, and the average person on the street would have, which is that some part of the brain lights up when you're scratching your butt or chewing gum or climbing a mountain or whatever cajillion things that you can be doing, there's always some kind of signature in the brain. This is a correlate of brain activity through measuring the blood flow in a specific region of the brain. You'll probably also recognize some of these images, microscopic images of the brain. Of course, you're only able to see a small part of the brain at any given time. And here is the meat that we're all familiar with as well, which does not belie its true beauty and complexity. Now, I'm coming at this from the thing that I'm bringing from within myself and from my own observations to the field of neuroscience art is my love for Japanese, Chinese, and Korean art in particular. And this is a piece from the Edo period in Japan called Red and White Plum Blossoms, one of my favorite paintings of all time. These forms have a lot in common with neural forms, these random branching patterns which have been painted in this tradition for a very, very long time. And my goal was to try to bring the aesthetic of this sort of Zen emphasis on asymmetry and spontaneity and negative space to couch the forms of neuroscience into a context that people weren't used to seeing. There's a deliberate attempt in these paintings to have the viewer say, well, am I looking at something botanical, or am I looking at something microscopic, or am I looking... These fractal neural patterns exist at many different scales, and couching it in the context of Asian art when you might have been kind of used to seeing these types of forms, I think is a good way to introduce people into the topic. So a lot of my early paintings were gold leaf and ink on paper, and then I started to branch out into more metallic media. This is a piece, Cortex and Metallic Pastels, where I'm using reflectivity to be changing the way in which the viewer perceives the piece. If you were to be looking at this live, those bright green stripes that you see here, these guys, that's actually from lights that are shining on the piece from overhead here. The background is etched in a horizontal dimension, and it looks sort of like a trout or like some iridescent snake skin or something like this that you may have seen before. And it casts these gold neurons into and out of silhouette. So these are my first experiments in micro-etching, essentially. Here's another piece called Spinal Cord. Again, I think the theme of my art is I like to draw parallels between natural themes that you see in different contexts. Here, I've driven some anatomists crazy by turning a cross-section of the spinal cord upside down, which some people, when they bought this piece, hang it upside down because they just can't stand that it's in a non-standard format. But in any case, it looks like an iridescent blue morpho butterfly and is illustrating all the inputs and outputs coming from the motor neurons here. A piece called Deciphering Spikes about multi-unit electrode recording of neural activity in the cortex here as being almost like aliens. We are the aliens abducting our own neural signals. Our brain has evolved to have the interest in understanding itself. That's what this piece is essentially about. Here's a piece called Hippocampal Formations, which is about the hippocampus, the region of the brain where we form episodic memories and complex sensory experiences in our brains, and is also the seat of learning and memory. And in this case was modeled after both the hippocampus and of the erosion patterns taken from satellite images over a desert in Jordan. And again, so these fractal forms continue to rear their heads in my work. Now I wanted to show you this piece, which is called Self-Reflective. This is a piece actually funded by the National Science Foundation and is my most complex work to date, without a doubt. What you're looking at is an object on the wall, too. This is not a digital reproduction of anything. This is not a scan. This is nothing like that. This is painstaking 2 years of work creating all the neurons by hand, scanning them to the computer, and doing a whole bunch of stuff that I'm going to tell you about in just a minute here. These will give you some closer looks. I'm going to fast forward it, though, to here. So this is self-reflected in animation mode, and the idea here is that we're trying to show you what the neural activity looks like from the perspective of an entire slice of the brain. This is the motor cortex that we're going into right now. This is where impulses are sent down into your spinal cord to help you move. We're getting into to frontal cortex in just a minute here. But the idea of this piece was to change the way in which the average person thinks about the brain, to marry the microscopic and the macroscopic, to bring something to the table which I personally had never seen before. My goal was not to try to replicate the work which is already out there. It was to break through the normal conventions of the ways of depicting the brain so that you could have a more visceral understanding of what the brain is. Here we're in frontal cortex right now. These are the neural correlates of logic and reason and overriding our base emotional response. These inputs here go into the basal ganglia and have some interesting effects. And so here, in the context of the larger piece, this is where we are right now. Unfortunately, I don't think there's time to show you this whole thing. I'll show you another bit of it here. This is the caudate nucleus, which is integrating essentially like decision-making and movement in the basal ganglia here. We're going to come into the thalamus, which was the central routing and gating center of the brain. Lots of inputs are coming and going out of here. And yeah, unfortunately, don't have time to show you more. But what the question really that a lot of people have then is, what is a microetching? So microetching was invented to be able to illustrate the brain's complexity in a different way, to be able to bring in a fourth dimension of data. Because what microetchings are essentially doing is they're using reflected light in order to animate something in a short loop. Self-reflected is basically like a hard-coded animated GIF, if you want to think about it that way. And the way that it's done is through etching millions and millions of tiny, zillions even, of tiny little etches in the surface of gold in this case. So this image right here on the right is an image of the microetching taken with a macro lens, so a very high close-up. And you'll notice that what's blue has a different angle of etch to it than what's green, than what's yellow, than what's red is going in this direction, picking up light. And the idea is that when the light is coming in perpendicular to the angle of those etches, then those etches will reflect that light, which also means that if you move the position of the lights, then all the colors change. Or if the viewer is moving around, then all the colors change as well. So motion of the light or the viewer will animate the piece. And this is what those little etches look like under a scanning electron microscope. On the right is a 1200x image, on the left 120x. All right, this is a little bit more deliberate about it. Let's say this is what the microetching data looks like before it's etched. Let's say we have a light source that we're going to move across the top here. And I'd like you to pay attention to this little region right here. Whoops, doing that. In fact, let's do this again. This little region here. See how at the end it starts as these etches going in one angle, and then as you move along this, by the time you get up here, the etches are going and picking up light from over here. So what's going to happen with this little dendrite right here is that you'll first see a spark of light here, which moves along this, and then moves into the soma here as the light source moves across. So this is the basis of the animation. There's a lot of math that goes into this that I'll touch on a little bit later. But this is how we have kind of invented this method. And I keep saying we. I have a collaborator, Brian Edwards, who's a applied physicist and engineer at Penn. Dr. Brian Edwards, I should say. So we decided that we were going to do an oblique sagittal slice because it would give us the most information. This is essentially what it looks like right here. There's a lot of cool circuitry in here. It's not a symmetrical slice, so we can show more things happening in it. And these are the main regions which are associated with this part of the brain. So we've got the cortex here, of course. This is the basal ganglia. Here's the basal forebrain, the brainstem, cerebellum, thalamus, etc. Now came the task of painting the neurons. And all of the neurons were painted by this technique, painted by breath, essentially. And the reason that we decided on this method is that your brain is terrible at generating randomness, which is essentially what a neuron is. It's a random branching pattern. Yes, there's rules and logic as to how it's growing into its environment to make connections with other neurons, but at its heart, it's a stochastic process, as is Brownian motion. There's an aspect of that to it, where molecules are just kind of bumping into one another at that scale, and it's weaving around other objects in its wake. So these were painted with this breath technique. There's probably 100 to 150 categories of different neurons and 5 or 6 examples of each. They were scanned into the computer, and this was done with some students who were fine art students, engineering students at Penn as well. Scanned into the computer, and then using vector art brushes, painted into the entire brain. This is just one small section of cortex. It's probably 1,000, 2,000 neurons. The entire piece has 500,000 neurons in it. So this is how we're painting down the gray matter, which are the actual cell bodies of the piece. But the other, I would say, arguably more complex part of this project was capturing the white matter. If you look at this stain on the right here, this is a sagittal slice of the brain. The black part on the right, all this stuff in here, is you're staining the myelin sheaths, which are the fatty coverings of the axons. The axons are, of course, carrying information from neuron to neuron within the brain. The question was, how can we depict the axons? Because they're so beautiful. There's just millions and millions, billions of these axons all threading through one another, bringing information to various places in the brain. So to that, we turn to my buddy, Dr. John Piles at Carnegie Mellon, who took some scans of his brain. Now, the scans are not usable directly for a variety of different reasons, but we nonetheless were able to get some pictures of his brain, both T1 MRI scans, which are looking more at structural components, and then this is diffusion spectrum imaging data, which is similar to diffusion tensor imaging, where you're using various methods to detect how water is diffusing through tiny little tubules in the brain. You get these awesome images. So now the axon tracks were all painted essentially by hand, and now we had two-dimensional data of all the neurons and all the axons, basically all the axons. Now, how do we put these together? That's really the crux of this project. Well, brute force, and a very sophisticated algorithm written by Brian to be able to essentially route information through the brain. The basic step of how neurons are communicating with one another is, we're calling it a choreography. We have, let's say, the thalamus is sending information to the motor cortex, which is then going between the layers of motor cortex and then going back down into the thalamus. Each one of these lines here is indicating one step of that choreography. And all of this information here is indicating how fast the axons are conveying information, whether or not the neurons are firing as a burst or individually, that sort of thing. So there's a lot of control, and there's about 600 or 800 lines of this, which are choreographing the entire brain. And so a lot of people, when you think, oh, artists are making some work about the brain, let's get a bunch of hippies together and have them throw paint at a canvas and see what happens. It was most definitely not this type of art. It was not so free-spirited. It was much more like just doing your taxes all day, every day. That's the thing with art too, is when you have specific constraints, when you know in your mind what you want it to look like, it oftentimes can become a process of manufacturing that goal. It's how do I put it? It's not as if you're robbing the spontaneity of it, but it's kind of like that a little bit, because you're beholden to an image. It's not so much, let's see how this turns out. It's, we kind of know what it looks like, but we're taking it a few steps further from where we're used to seeing. I'm going to skip this for now. I don't think we have to. Well, actually, I'm going to talk about this one specific point. All right, let's say you are this neuron right here, and you're receiving inputs from these 3 neurons here. In order for this neuron to fire, it has to receive inputs at the same time. That's how neurons work sometimes. The cumulative input of its connected neurons will control whether or not that neuron fires. So if you think about this in terms of time, this neuron, because it's the farthest away, has to fire first in time in order for its impulse to arrive at the same time as these other two. So essentially what we're doing with this algorithm is we're sequencing things in time. And in the ensuing images, time is going to be represented with color. And I apologize to you who may be colorblind or otherwise unable to perceive these next images. This is a map of time. The red pixels would be the pixels that you would see when you're standing all the way to the left of this image, when it's illuminated. Okay, this is just a data map. This is not what's hanging on the wall, essentially. This is the input data, which is going to go into another algorithm I'm going to tell you about. So when you're standing all the way to the left, you see the red pixels. Then when you move to the right, you see the orange ones and the yellow ones and the green ones, blue ones, violet ones. So you can kind of see how information is moving through this piece. And if we zoom in a little bit, this is what it looks like at high res, and it's a mess here. So let's look at just this axon right here. We're starting with red, and it's becoming increasingly more orange as you get over here, meaning that information, that a little dart or action potential is going to be moving across this axon as the light moves. How do we make that happen? How do we turn this color image into an etching? Well, first we need to know where the light source is. That is the absolute heart of microetching. You cannot make them without it. You cannot make it accurately if you don't know its exact position, etc. Let's take some pixel on the surface, just one in the middle, let's say. And we know what the relationship is between that pixel and the light source, what angle the light is hitting that pixel at, because we know where the light source is. We also know, based upon the color of the pixel, where the viewer is going to be at that moment. Remember, if the pixel is orange, the observer is going to be a different place than if it's green. Okay, so now we know those two vectors, those two directions here. Now we can calculate what angle of mirror you need to make at that pixel to reflect the light from the light source into the viewer's eye. Now what we're making, pixels are square, right? They're not these flat facets. So what we're truly making is a little grid of cylindrical reflectors. Cylinders are great in this case because you can see them if you're tall or you're short. If these were facets like a diamond, the reflections would be brighter and sharper, but you'd only be able to see them at very small areas. And they're really, really hard to make. So this is what each pixel looks like. So we calculate this for every single one of the pixels on the entire piece in order to get an image that looks like this. You can kind of see, here's a cell body right here. You can kind of see that the neurons and the axons are now all defined by their level of, or by their direction of etch. That should say too, this is zoomed way, 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 way in. This is probably 15 to 20 times higher resolution than your printer at home can print. Okay, so we print all this data onto a transparency using these really specialized printers, and this is what the transparencies look like. And then we do photolithography to actually make the physical etching. That's what we're doing now, physical etching. We've got the transparency, and we're going to now lie the transparency down on a flat on this surface that we're looking at in cross-section. Here the substrate's just a piece of steel or acrylic. It doesn't really make that much difference. And then the yellow part is the photoresist, which is a photosensitive film that's laminated to the surface. Then the the transparency is put on top. Then you bake it with a ultraviolet light. Now anywhere where the transparency is clear, that light penetrates through and it polymerizes a spot on the photosensitive film. It makes it tough. But the black printing is so opaque that it blocks the UV light from hitting that, so it doesn't get exposed underneath it. So you burn the image in all at once in parallel. This is not an etch where you're using a laser that's scanning over the surface or anything like that, or a mechanical etch where you're scratching it. You're using film and chemicals and light in order to engrave this. this very fine surface. So you turn off the light, remove the transparency, and then you, from this step, you dip it in a solution of a weak basic solution, which eats away the parts that were unexposed, leaving only the hard parts. So anything that was under the black printing gets dissolved away. This is what the setup looks like in my studio. This is the plate that's exposing right here. This is the clean room where we're making the etchings. Here's the UV light source right here. And then I'll tell you, right. Okay, so we're putting gold on the surface here now. I forgot I didn't put a slide in about this. This is right here the purple plates. That's what the photoresist looks like after it's come out of that process I just described to you. And now we're putting gold on the surface. These are two of the assistants, Becca and AJ, who are helping me with this project, where single sheets of gold leaf are applied to the surface and burnished in to increase the reflectivity. The entire point of the art is that it's reflecting light, so you want it to be as brightly reflective as possible. So we developed some methods to be able to do that. And gold leaf is bloody perfect because it's gold and everybody loves gold. And it's so thin and malleable and soft that you can cram it into all that topography that you've just made so painstakingly without it changing that topography appreciably. In other words, the math of how the etches are rounded, how the angles are situated, all that kind of stuff is so specific that if you put anything on it which is thicker than like 200 nanometers, which the gold is, very, very, very thin, then you run the risk of distorting the image or making it invisible or not having enough clearance for the light to scrape down across it and get to your eyes. There's a lot of things that can go wrong. So this ended up being really the perfect solution to it. And this is the piece self-reflected under a single white light. This is the full-size version of this is hanging at the Franklin Institute in Philadelphia, and then I have smaller versions of this available as well actually on my website. But remember, we're talking about a reflective template. Color is an infinite variable. This is what the same exact piece looks like when you just have different color lights on it. Now this has a purple light and a pink light and a white light on it, and all these colors change as you're moving around. It's pretty cool to watch. So here's a close-up of the thalamus and the basal ganglia right here. Here's the nucleus accumbens down there. The cerebellum, one of my favorite regions. All these Purkinje cells sending their axons into the dentate nucleus here. And there's just a zillion granule cells here, 50 billion of them, which comprises about half, probably more than half, of the brain's total neuron count. This is a close-up of the parietal cortex, which illustrates pretty well how the cortex is oriented into these neural layers. And also the color here is informing you what is functionally connected, what's firing at the same time. All the blue neurons are firing at the same time. All the red neurons are firing at the same time. So that color coding really does give you some insight into what's happening. Here's the cortex, excuse me, the motor cortex here and the somatosensory cortex here. This is your brain processing your sensory inputs. The brainstem and the cerebellum here. And this is that big thing at the Franklin Institute here. This is at the opening. And this is my collaborator, Brian. So we were extremely fortunate to be able to do this project and to get funding to do it too. We're grateful to the NSF for the opportunity to be able to do this. And obviously this project was a lot of effort, and it was a lot of inventing new things and a lot of troubleshooting things which didn't work. And there were a bunch of times when in the process I was just asking myself, why? Like, what is the point of art? What is the point of all this? You know, as an artist, as a professional artist, I get lost in that rabbit hole actually quite often. And the thought that kept bringing me back to it was imagining a kid walking up to this thing for the first time, not really knowing anything about the brain, and standing in front of this massive golden glittering object and have the light go over it for the first time and to have the brain explode into the activity of 500,000 neurons at once and for that kid to have that one moment of just, wow. For anybody, for a professional neuroscientist, for the general public, the whole point of this piece is just to refresh ourselves, to refresh our memory that our mind is a miracle. It is, if I scream at you, the brain has 86 billion neurons. That means nothing. That means nothing to the average person because we have no context to understand what that is. Clearly, in order for something to give us the degree of sophistication that we have in our daily consciousness, it's got to be very complicated, but communicating that idea is very challenging. So the point of this was to allow the viewer, using their own senses, to see 500,000 things in activity at once. To get just a tiny slice of that complexity and to feel it with their own emotions. That is the most important thing about any piece of art. What do you feel in your emotions? I think it was Maya Angelou who said, you may not remember what I said, you may not remember what I wrote, but I'll always remember how you made me feel. Forgive me, I butchered the quote. But the general idea is that it's our emotions which are driving the ship, more than our logic, more than our senses. In order to truly make an impact on somebody, I think addressing that idea is crucial, and it's something that I really take great pains to do in my art. So that's what I wanted to tell you about this piece, Self-Reflected, and I wanted to talk a little bit about, I guess, how I've come to this place. And again, it was never the intention. What did I want to be when I was a kid? I think I wanted to first work in a gas station, and then I wanted to be a rock star, and then I wanted to be a vet. Actually, that's what I wanted to be for a long time. I studied biology, more kind of like gross organismal level biology. Then I wanted to be a molecular biologist. Then I wanted to be a neuroscientist and a professor, and somehow I ended up here. And I think that what brought me to this place was, I guess, the integration of a lot of experiences in my life. That's what's going to give people their most unique voice here. So if I'm saying always seek new information and inspiration, the more stuff you've got going on in here, the more your brain kind of does its magic and finds connections between things. And that's really what the individual has to offer. There's a lot of people who are interested in neuroscience. There's a lot of people who are interested in Asian art. There's a lot of people who are interested in reflective surfaces. There's not a lot of people who are interested in all those things together. So having a broad base of interests, I'm more kind of a jack of all trades kind of guy, I guess, and I enjoy finding connections between things in that sense. Going outside your comfort zone is also extremely important, and is something that I need to be reminded of all the time as well. Where, you know, the way that the world has changed is by changing paradigms and dynamics, not by doing more of the same, not by doing yet one more brain illustration of the transparent brain in electric blue. We've got enough of those, and I think that making a lot of work helps for that to happen. It's particularly work that you're not concerned with other people seeing. That is so important. Like, part of being a good artist, in fact, a lot of being a good artist is just knowing how to edit your own work. What to show and what not to show. What secret experiments that you do in your studio that are horrible, that nobody should ever see, where you feel the freedom to be able to experiment. And making work really helps you to, I think, kind of hone your voice. And of course, as you hear from many, many different spheres and disciplines, failure is a part of the process. You're not going to get anywhere without failing. And as many have also said, simply like failure, giving up, not even giving up isn't even failure. I would say that that's another thing actually that's important. Failure can also be defined as putting too much effort in something which eventually won't work anyway. Um, I think that this is part of making work, is iterating your sense of what's going to work, what's not going to work, what's going to be appealing, what's going to change people's minds, what's going to, that's, I think, failure is difficult to define in some ways, whereas honing your ability to edit yourself is maybe a better way to describe that. So, as I mentioned before, if you're looking at the Venn diagram of the things that I like to do, a couple things here: Asian art, brain and mind, making new types of images and sparkly gold things. What's at the center is what I try to do, and I've got some things which are obviously in other compartments of this Venn diagram. But in making science art specifically, the Venn diagram that I found that works best for that is obviously that the scientific content has got to be solid. It's got to be there, and you've got to do your research in that sense. If you're trying to illuminate for the average person what a tricky scientific concept looks like, it's got to be emotionally engaging. If it's not emotionally engaging, if you don't give people a reason to care about what you're doing, nobody will care. The internet is full of millions and millions and billions and trillions and quadrillions of things for you to look at. There has to be some kind of emotional hook to it, and it has to be a deliberate part of the process, or it's going to be a struggle to come up with something that people don't, or that people actually are paying attention to. And for example, you have an artist who's painting sunsets and is upset that they're not as successful as they think they should be. The answer in a lot of cases is, unless you are one of the 10 best sunset painters in the world, there are millions and millions of people who are painting that. You haven't defined your voice, perhaps, as specifically as you can. And that feeds into the aesthetics and style. What do you bring to the table in terms of how you prepare your images? How do they look different from everybody else's? And then the conscious awareness and rejection of convention. I tend to be the kind of person who finds it easier to write haikus than free verse. Like, I like having constraints that I'm working under. So if I have gone through grad school and I've seen 100,000 different images of the brain, I know how it's been typically depicted, and I'm looking for the thing which I haven't seen, which in my mind is describing what the brain actually looks like. In other words, I'm looking at the illustrations which are there, and I'm consciously departing from them. And if all these things work together, and they for sure don't all work together for me all the time, then at the very center of that's where you get those moments which truly change people. If you want to know more about my art, I've got a website, gregadunn.com. There's a bunch more paintings here on that site for what I'm doing. I've also got some social media stuff going on. And I also wanted to take some time now to talk about a little bit about my career and how I got to this point, and what motivates me, that sort of thing. I've just got a few questions here that some folks had submitted that I wanted to go over here. In terms of how I got my start doing this, so as I mentioned, I was in grad school and I had really wanted to become a professor. And I started painting the brain maybe a year into my tenure as a grad student. Because I needed some damn experiments that worked for a change. That's something I think the general public just doesn't really grasp very well, is that academic bench science is complicated. It's hard. It's pretty hard. And the results you get are almost never 95% efficacy, like the miracle Pfizer and Moderna vaccines. You almost never get that kind of thing in the lab. So, when you're toiling away on your experiments, sometimes for months, and the data isn't clear, the experiments aren't working, whatever it is, it was nice to be able to come home at the end of the day and make a thing that I could show to people, you know, as opposed to just having this ethereal discussion about what transgenerational epigenetics is, and that's what I was working on in grad school, is um, how epigenetic traits, basically the environmental factors which change how our genes are expressed, pass through generations. So that's why I began painting. I like to have a thing at the end of the day, and it was a way also for me to process the incredible opportunity that I had as a grad student, just to be learning so much about what is, in my opinion, basically the most fascinating thing in the universe, is the brain, all brains. Um, so it was about, and I started selling some pieces. I put some work together on a website and that kind of thing, and it was nice because my audience was already very clear who I was making work for and why. And I was commissioned by the Society for Neuroscience to do a large gold leaf painting for their lobby, which was amazing because it was basically them paying me to put a giant billboard in the best location in the universe, uh, as an advertisement for my art. And that's a big advantage that visual art has, by the way, over other art forms, is that you can consume an image almost instantaneously when you see it, you know, as opposed to an author writing a book where somebody needs to dedicate, you know, 5, 10, 20, 50 hours to reading it. It's a more immediate um, communication. So I felt that by the time I finished grad school that I had developed my voice enough as an artist and had enough work too, that that my wife and I kind of discussed it for a while and decided that, what the hell, why not? Let's give it a shot. I'm gonna try to be a professional artist for a while and see how that goes. Because I didn't really feel like I was doing anything in the lab which was something that others couldn't have done. Yeah, I was fine. I was reasonably talented in the lab, but I didn't feel like I had my voice as well carved out there as I did when I was making images of the brain that were communicating both to professional neuroscientists and to the lay public. That seemed to be more my calling in a sense. So that's how I got into the career. You know, I'd set up a bunch of commissions and things so that I could hit the ground running as soon as I finished grad school. And it was scary. You know, your whole life, people are telling you, don't be an artist. There's no money in it. You know, if you want to support a family, it's going to be very difficult. And that hum. I mean, to this day, I've been doing this for 12 years now. It doesn't really go away. I still have that fear in some way, even though that I've been grateful to be successful at this. So it took a while to get over that, to feel like, you know, I had some solid ground to stand on, to feel like the next job is coming, because it's a very different thing to be. I essentially went from being an employee of a lab to having my own lab, my own art science lab, where your productivity is not measured in what you, like, the number of hours that you showed up. It's completely different. It's how much have you actually produced today. That's the only thing that matters now, which is a very different perspective to be in coming out of grad school, and is one which I think is incredibly valuable to have experienced. So, I mean, I think that the main lessons that I've learned along these lines are, if you're going to do something like this, you absolutely must have a voice that is unique and that you know where your audience is. Because in order to make a living, you have to be able to make money. You got to be able to sell your pieces, and knowing what your voice is, and then making the absolute best work that you possibly can, so that it is its own advertisement, is not a bad thing either. So be unique and do that unique thing as best as you possibly can. I particularly like working at the intersection of art and science, because my brain works both ways, and there's so much to be gained from either side. You know, art pure fine arts in the high concept sense, I oftentimes find extremely frustrating from the sense of how is this communicating to somebody who doesn't understand all of the oblique references that are being made in this obscure piece of art? What is the immediate kind of transmission of information to the person looking at it? I feel like there's a lot of artists who don't take that into account very much, and I find that frustrating. And my philosophy is different. And I'm sure that somebody would argue with me about this, and that's fine. But from the science perspective, it's very easy to lose that sense of how how am I doing anything other than just communicating dry facts to people? There's just a beauty and wonder of the brain, or of just science in general, that can be so effectively communicated through art, that I think that the combination of those two things really brings the best of both worlds, to be able to get scientists to appreciate art and artists to appreciate science. I don't know. I'm the middle child of my family, and I'm always trying to kind of bring people together. I guess that's kind of my role in life. So that's where I ended up. What else here? Art make neuroscience. How does art make neuroscience a more approachable and inclusive field of study? Well, I think for some of the reasons I was just mentioning, that usually the way in which we interact with kind of scientific, neuroscientific information that we hear about on TV or stuff like this is either through pop science or through stuff which is too complicated, or than the average person is willing to put the time in to be able to understand. So the arts, again, my goal as an artist is to be able to make it accessible enough. Is to give something to people that's beautiful to look at and encourages them to ask deeper questions about what it is, and I think to better appreciate the sacredness, I would say, of our brains, the part of us that is at our most basic foundation. In terms of some of my biggest influences have been Jim Eberwein. He's a neuroscientist at Penn. He was a big influence on me, as well as Ed Boyden, who's at MIT. And I like both of them because they use an art science approach themselves, actually, now that I think about it, where they're both incredibly talented and intelligent people. They study different things. I'm not going to get into that. It doesn't really matter at the moment. But what I like is that they are synthesizing their scientific pursuits with artistic ones, in that they're constantly inventing new methods in order to figure out complicated problems. And that's where the art comes into it. That's where the synthesis of creative ideas in order to solve a hard problem whose constraints are well understood. That scientific data tells us what we need to know sometimes. How can we figure out a way to solve what we need to know? I have a lot of favorite painters. I'll mention just one, Ito Jakachu, who's a Japanese painter from kind of one of the eccentric schools. If you're interested to look him up, Jakuchu starts with a J. And he's got this really great eccentric style that I like a lot of spontaneity, a lot of love goes into his paintings. And then I guess finally, if I had a favorite work, what is it and why? I'm going to give you the annoying dad answer and say that I love all my paintings equally, which is not true, actually. I don't love them all equally, but there's things that I like about certain ones and not others. But Self-Reflected, I think, is my greatest achievement, just because I think it breaks the most boundaries and just has a crap ton of data in it. And I think has had the greatest reach of any of my work. Its goal when I first started it was, again, to change the way in which the average person thinks about the brain. So I think that's the one that I'm the most proud of. Well, thank you very much for listening. I appreciate it. Wish I could be there in person, but I'm very, very far away from you right now. And let me know if any questions come up. Please feel free to send them to me via my website. And yeah, thanks again for your attention. Take care. Bye.
Thank you so much, Dr. Dunn, for your wonderful presentation. It's an honor to host such a distinguished artist, and Gray Matters is thrilled to be able to share your work with our audience.
Thank you everyone for coming. Please take our feedback survey at tiny.cc/EWN feedback.
We hope to see you next Friday at 5 p.m. for our final event, the panel discussion and live Q&A. You can submit questions for the first half of the discussion at tiny.cc/EWNask. Question submissions close tonight at midnight.
Thanks everyone, and enjoy your Friday evening.